So thank you very much for this invitation. I'm very excited to be able to show you some of the work that we have been doing in my group in the past few years. All the work that we do is computational, as you will see. And one of the main ideas of our work is to try to link the chemical diversity of molecules and uncover molecular and biophysical mechanisms that might explain how this chemical makeup of the molecules gives rise to complexity in biology. So how the molecular interactions give rise to self-organization that has some functional implications. And the example that I'll be discussing with you today is our investigations into chromatin organization, again, from the point of view of biophysics and molecular mechanisms. So one of the main interests that we have in general is to develop new computational and theoretical approaches that allow us to gain these biophysical mechanisms. And from the point of view of genome organization, this is a field that has gained a lot of attention because of many reasons, as you might know. One is that genome sequencing has become so efficient at this point that it can now be used as a routing tool, which has allowed more emphasis to be generated around the idea of not only how do we sequence the DNA, but also how do we interpret the functions and the information that is encoded in our genome. So this, as you probably know, is a very difficult problem to solve because of different reasons. One is simply that DNA is a huge macromolecule. If we were to stretch the DNA in each of our cells, we would see that it's over 2 meters long, and it has to undergo dramatic condensation in order to fit into the tiny nucleus of our cells, which are almost 1 million times smaller. So secondly, this condensation process is a highly complex process. And just to put it in context, something that is, I think, a very powerful analogy is this idea that this condensation is equivalent as taking one of our hairs, which is super thin, but imagine a 20-kilometer long human hair and trying to compress it and fit it inside of a golf ball. And not only that, which would be dramatic decondensation, but also the cells face the problem of trying to search for specific regions of our hair while the hair is inside the golf ball. So it's a very, very hard problem. And on top of that, organized, like folding a piece of paper in different ways, like I have in my example here, to give rise to, let's say, a bird or a boat. This organization of our genome inside cells seems to be varying across different cell types. So in a way, in very, very simple terms, it has been suggested that these different ways of organizing the same DNA information might help or might contribute to the way in which cells acquire their identity from a mechanistic point of view. So this is the motivation that we have in my group to try to understand how these different ways of organizing the same DNA information emerge and understand from the biophysical and molecular point of view what shapes these different ways of organizing the DNA and what maintains these different organizations. So in order to begin to understand this question, let me give you a little bit of context about what we know about genome organization from the very detailed chemical and molecular point of view. As you probably know, the condensed genome is known as chromatin, and if we actually had techniques that allow us to zoom in into the condensed genome, we would see that the DNA is not found free in eukaryotic organisms. It is always complex with histone proteins, with an octamer of histone proteins forming these very important nanoparticles known as nucleosomes. Now, nucleosomes have some very important properties. One is that they have 10 intrinsically disorder histone tails, which allow the nucleosomes to start acting as a smart glue. So these intrinsically disordered regions are positively charged. They can contact distant DNA regions or immediate nucleosomes and begin to initiate this condensation process and also begin the organization process. So how can these histone tails and nucleosomes begin this process of organization? Where here's where epigenetics comes in play, and especially this chemical diversity that I was talking to you about that we are very, very interested in my group. So again, if we were able to zoom in into the condensed genome, we would see that nucleosomes, and in particular this disordered histone tail that I just mentioned, carry an extra layer of chemical information. So an additional chemical code on top of the genetic code known depending on how you define it as the epigenome. From our point of view, we are interested in this epigenome because it contains a lot of different chemical groups like phosphorylation, methylation, acetylation, and also a wide range of additional proteins that are recruited specifically or non-specifically to these different chemical marks that are distributed along the genome. One key point from the point of view of functional organization is that these different chemical marks are not distributed homogeneously across the genome, but they form different domains. Although some of these domains do have overlapping marks, some of them are enriched in certain combinations, and some of these domains are enriched in different combinations of these chemical modifications. And it has been suggested that these different combinations of marks might explain the emergence of the functional units of the genome. So let's say some combinations might give rise or might explain why active domains in the genome emerge, and some combinations why inactive domains where genes are off originate. Now, as I mentioned, we are interested in the chemistry and biophysics of why these things happen. And at the moment, we are not able to understand how the organization of these biomolecules and within these regions transform the way nucleosomes interact with one another, or the organization of nucleosomes within these different regions. So this means that we are not really able to rationalize how chemical modifications lead to the reorganization of the genome at scales that are 3 to 4 orders of magnitude larger than the sizes of these molecules, which I think is a fascinating multiscale question. And one of the problems here is that solving the organization of these small 10 nanometer nucleosomes that I mentioned within very dense micron-sized structures is a very challenging problem. And one of the problems, for instance, is that these 10 nanometer particles lie within the blind spot of standard imaging methods. So this has resulted in chromatin organization mainly being resolved at 2 limits. One is the limit that I'm showing you here at the left corner, which is very high resolution, atomistic, near atomistic resolution, but with relatively small system sizes, although these systems are growing significantly at the moment. Or the region of systems that are larger than the sizes of genes and therefore have functional relevance. They allow us to understand how function relates to a structure, organization relates to function, but with relatively low resolution at the moment, so coarser resolution than single nucleosomes. This means that we still, again, cannot see how these small molecules are affecting the organization, the interactions of nucleosomes, and giving us a blind spot that explains exactly how this chemistry relates to organization. So there's many experimental techniques, I must say, that are actually advancing enormously at the moment, including electron tomography, cryo-EM, super resolution microscopy, that are all actually pushing at the boundaries and getting very near to the blind spot. So, however, all these techniques rely on computational modeling to predict the structures that match the experimental observables, for instance, or that are consistent with experimental observables, which means that all the inaccuracies that we have in the computer models can be transferred directly into the interpretation of the data. And this is where our work comes into the picture. At the moment, one of the main sources of error in modeling chromatin structure at high resolution is the neglect of the heterogeneity of chromatin that I was mentioned initially in terms of composition, chemical composition, but also intrinsic factors of chromatin itself. Our previous modeling work investigated one of these intrinsic factors, which is simply the length of the DNA connecting successive nucleosomes, which varies across within the same chromatin system inside cells, across tissues, as well across cell types. So if we treated chromatin, as I'm showing you here on the left, as a homopolymer where everything is homogeneous, all the nucleosomes are the same, all the DNA sequences are the same, all the DNA connecting successive nucleosomes are similar, the same. We observe that chromatin forms the archetypical rigid, very ordered zigzag fibers that you can see with in vitro experiments, also of small systems with similar like regular parameters. However, if we just vary something very simple, the length of the DNA connecting successive nucleosomes, we already see a significant transformation in the conformational ensemble of chromatin from these rigid fibers to a polymorphic organization. Suggesting that this heterogeneity can transform the predictions significantly. So what we are doing in my group at the moment is trying to bridge different scales with our models. So bridging atomistic descriptions of nucleosomes, proteins, and DNA to get an idea of how chemical modifications transform the local environment and the interactions among nucleosomes and proteins and nucleosomes, with different levels of approximations that are called coarse-grained models, in which now we represent many atoms with a sphere or with an ellipsoid with different objects, but still maintain the chemical identity of all the species within the system, so that we can investigate how protein sequence effects or DNA sequence affect organization at large scales, even to the limit of phase separation, as I'll tell you in a few minutes. Now, we tested this type of methodology earlier in 2015 to try to understand from the molecular point of view the very well-known dramatic decondensation of chromatin upon H4K16 acetylation. And again, as I mentioned briefly in the previous slide, we combined atomistic simulations of proteins, of histone tails, with and without the modification, and also of dinucleosomes, interacting nucleosomes also with and without the modifications with NMR data. And we observed that this modification, besides simply inducing a charge reduction of the H4 lysine, also induce a rigidification of the histone tail, increase the content of secondary structure within the tail, effectively making these histone tails unavailable to extend and contact neighboring nucleosomes and inhibiting nucleosome-nucleosome interactions. When we map this into our coarse-grained models, we observe that indeed this rigidification, as I'm showing you here on the right, explain the unfolding of chromatin, whereas if we only consider charge reduction, we still could observe fairly condensed systems. So this result, the interesting part of this result, I think, is that it suggests that the effects of post-translational modifications of proteins on histone tails are not trivial and need to be investigated at a high level of resolution in order to be able to map the large scale effects mechanistically. So doing something similar, again, with multiscale model, we investigated more recently how binding of a highly disordered protein to chromatin influences its structure. We looked specifically at the H1 linker histone protein, which is one of the most abundant proteins within heterochromatin silent regions. So we use, again, atomistic simulations now combined with an enhanced sampling method. And we observe that the long long terminal tail of this histone protein, the C-terminal tail, remains disordered actually when this protein is bound to the full nucleosome particle, which is something that had not been observed, but is consistent with experiments of the H1 C-terminal tail bound to DNA, and also of H1 bound to a long negatively charged IDP. So more interestingly, our simulations also reveal that the disordered nature of the C-terminal tail of H1 increases significantly the flexibility and the fluctuations of the linker DNA that connects successive nucleosomes. And as a result, binding of this disordered protein actually destabilizes the very regular zigzag configuration that had been predicted that was promoted by the same protein. And this is something that is consistent with experiments as well, more recent experiments. Okay, so now to try to get closer to mimicking, at least in part, some of this enormous complexity of in vivo chromatin, we are addressing a few more features of chromatin in our models. So besides this chemical heterogeneity of the protein and the DNA and binding of proteins to chromatin, we are also investigating a key observation that nucleosomes themselves are highly dynamical and heterogeneous particles. That is that thermal fluctuations can cause constantly some of these DNA histone core interactions to spontaneously break and reform in a phenomenon that is known as DNA breeding or nucleosome breeding. It has different names. So from the modeling point of view, this is something quite tricky to capture, but my student Steve recently developed a coarse-grained model of chromatin that has chemical specificity, meaning that is sequence dependent in terms of both the amino acids and the DNA. And importantly, does not force the DNA to be permanently bound to the histone core. So this binding emerges spontaneously just from electrostatics, hydrophobic interactions, and also very, very importantly, the sequence dependent mechanical properties of the DNA. And the results of this model are in quantitative agreement with mechanical unzipping experiments of nucleosomes, which tells us that the way in which we have parameterized the model is adequate. Okay, so now when we use this type of models to investigate the behavior of chromatin with nucleosomes that can breed, that are not static disks, we see that chromatin does not form these rigid zigzag fibers, but instead we have a system where the nucleosomes remain mostly intact as you are seeing in the movie, but chromatin has an impressive flexibility, which the ensembles that we are seeing are consistent with the idea that chromatin populates this a wide range of structures, like in this idea of the liquid-like behavior of chromatin, which is termed in this way because of its high structural variation, its disorder. So in more detail with our models, we can get more information about what characterizes or how the structure can be characterized. So here we are looking at how the nucleosomes are organized within chromatin with breeding nucleosomes here on the right versus a control case in which we now have forced the nucleosomes to remain intact. So there's no breeding, their nucleosomes are perfectly formed all the time. So here in these figures that I'm showing you here, we are plotting the frequency of interactions among first nearest neighbors and second nearest neighbors, third nearest neighbors, and so on. And we are breaking down these frequencies of interactions in different orientations of the nucleosomes, again, to capture some of the order behind these structures. So here in the control case where nucleosomes cannot breed, we see that most of the nucleosomes are interacting most of the time only with the second nearest neighbors and always organized face to face in the blue bar here, or most of the time, which is the archetypical zigzag organization of chromatin. On the left, on the right, we see that in huge contrast, as I showed you also in the previous movie, when there's DNA breeding, chromatin instead has nucleosomes form a wide range of different interactions, and they can orient themselves with respect to one another in a wide different range of forms. Okay, in addition to like this diversity of like single structures, we can also monitor the dynamics of nucleosome interactions in our simulations simply by looking at how often two nucleosomes that are bound together remain bound, and we represent this with a value of 1, or when they are unbound and represent that with a value of 0 and measure like whether they are bound or unbound versus time. And again, we see here on the left in our control case where we have forced the nucleosomes to not breathe, that basically the structures are very rigid. Once two nucleosomes are stacked on top of each other, they are bound. They basically remain bound most of the trajectory. And in huge contrast, just by allowing nucleosomes to breathe, we see that now the interactions are very dynamic. So the nucleosomes bind and unbind constantly. Again, consistently with this idea of the liquid-like organization of chromatin. In addition, from like the more interesting like chemical point of view, our models can also assess the detailed molecular interactions that stabilize the different types of structural behaviors of chromatin. In this particular case, we are contrasting, again, breathing versus non-breathing nucleosomes, but we can also look at chromatin with different chemical compositions. And for the sake of time, I'll skip most of the details, but I'll only tell you that we observe that these order fibers with non-breathing nucleosomes are sustained by dominant histone core interactions, the globular domains on the face-to-face interactions, and also some H4 tail interactions, which is something that has been observed experimentally for zigzag fibers. Whereas the liquid-like behavior of chromatin with these breathing nucleosomes is instead assembled by transient promiscuous interactions that involve mostly the flexible disorder histone tails at the DNA and the DNA, sorry. So no histone core interaction, just DNA histone tail interactions. Yeah. And importantly, we also find that this nucleosome disorder that I was mentioning within chromatin with breathing nucleosomes increases the valency of nucleosomes. This is important for phase separation. I'll mention it in a second. So that means that each nucleosome binds to more of its neighbors per unit of time and volume than nucleosomes within zigzag fibers. And we also observe that we get, this is a measurement of the sedimentation coefficient, so how condensed the different fibers systems are. And when we allow nucleosomes to have these spontaneous breathing motions, we see that chromatin can pack, can form more dense systems, so can pack its contents more efficiently. So overall, what I've just shown you confirmed that there are ways in which chromatin can be stochastically folded and also very condensed, so tightly packed. And also that having a very condensed organization does not necessarily immediately imply a steric varying of enzymes. So the DNA information in a compact system can also be accessible. So one of the main ideas that attempts to explain how chromatin might be organized in a space inside the nucleus so efficiently to control accessibility, and that does not rely in this picture that a rigid fiber-like structure is needed to explain gene silencing, is the idea of liquid-liquid phase separation, and that's why we are interested in this idea. In very simple terms, the idea is that within the cell nucleus, chromatin, as you know, is immersed in a very complex biomolecular environment, and it is in contact with a highly multi-component mixtures of proteins, and many of these proteins are multivalent. That means that they can form many connections, more than 3 connections with other molecules at the same time, which allows them to form the kind of network connectivity that you need to sustain a liquid structure. So above a certain critical concentration, these multivalent biomolecules can form many weak transient connections and lead to the mixing phase separation. The field of genomic liquid-liquid phase separation has exploded in the past 3 years, and there is now a lot of papers coming up in the field and evidence suggesting that this mechanism has many potential different potential functional implications. Some of the most famous examples that probably you've heard are the phase separation of HB1, which is associated with the formation of constitutive heterochromatin, silent chromatin. And at the other side of the spectrum, also, for instance, phase separation of transcriptional coactivators, which is suggested to enable the formation of super-enhancer regions or transcription factors, and many examples in between as well. And something very interesting from the point of view of our research is that it seems that the characteristics of chromatin, small chromatin fibers themselves, also make them suitable for phase separation. It was shown recently that chromatin arrays, 12 nucleosome arrays, can undergo liquid-liquid phase separation at physiological salt. And this has again gathered a lot of attention. So with our models, we are trying to get characterized again from the biophysical point of view, why does chromatin is able to phase separate, and how can phase separation of chromatin be modulated intrinsically. So we again do our multiscale techniques, bridging atomistic information of nucleosomes all the way to minimal models. And with these techniques, we are able to perform simulations of chromatin phase separation while resolving individual nucleosomes and considering nucleosome breeding too. And what you are seeing in this movie is a cross-section of a droplet. So if you were to take one of these liquid drops and cut it in the middle, you would see something like this, a condensate surrounded by a less concentrated liquid in chromatin. We validated this our models by comparing with the predictions of the Rosen lab on phase separation. So basically what we can compute is phase diagrams that are these graphs here that allow us to delimit the range of parameters, in this case, salt concentration versus chromatin density, and draw these curves that are, I'm showing you here, that are called coexistent curves or binodals, which basically separate the set of parameters that can stabilize a phase separation, which are all the space below the curves, or from the space where the system would naturally not phase separate, just form a homogeneous liquid, which is all the space above each of these curves. So these are different curves for chromatin with different parameters, in this case, different lengths of the DNA connecting the successive nucleosomes. And for chromatin, where the DNA follows this 10n+5 spacing, we see that chromatin phase separates very readily, so the size of the coexistent regions are larger, whereas in the case of the 10n spacing, where chromatin forms more ordered fibers where face-to-face interactions are favored, intra-nucleosome interactions within the same fiber are favored, and the interactions with other fibers are limited, and therefore the system does not undergo phase separation. So this is not new, this is just a validation of our models, but we agree perfectly with the experiments. And the more interesting aspect perhaps is that—
Rosana, I'm sorry,
yes?
We are about at the end of the time and we have many questions coming from the audience. So I wonder whether we should probably conclude and take a couple of questions.
Yeah,
are you okay with that?
This is actually my last slide, so I think yes. Okay, perfect. Okay, so I'm about to finish. Basically, what we observe is if we repeat the same experiment of computing phase diagrams, now testing the impact of nucleosome breeding versus non-nucleosome breeding, we again observe that these deformations of nucleosomes promote the phase separation of chromatin. And that is because of the same reason that I mentioned earlier about nucleosome breeding increasing the valency of nucleosomes, so promoting more interactions of each nucleosomes with its partners, and also because it allows chromatin to form a more densely connected liquid. So now we have a network emerging from these nucleosomes that can breed that is more densely connected. Okay, so in summary, I wanted to show you how we can use multiscale computational methods to try to link atomistic effects of nucleosomes to large-scale organization of chromatin, including phase separation, and what we can do to begin to capture some of this complexity of chromatin in vivo. Okay, so this is just to thank my students and postdocs for their fantastic work, our funding, and our collaborators, and I'll be happy to answer your questions.
Thank you very much, Rosana. This is super interesting, the power of molecular dynamics, and also this concept of phase separation, liquid-liquid phase separation, which has been applied just to proteins, right? So it's fantastic. And we have many questions, actually, so we will at least go through some of them.
Hi, Rosana, great talk. So we could sort of maybe keep the answers short. There's quite a few questions, and I'll try to ask as many as possible. So Zvabo Gabor says, thanks indeed for the highly interesting talk. I wonder how you take the ionic milieu into consideration. Potassium is not equivalent to sodium, and its effect on nucleosome stability dynamics features. It was reported by Kornberg earlier that anions seem to be much more important in the respect than cations.
Yes, this is a really good question. So we do this again with our multiscale techniques at the very coarse grain level. We cannot capture directly the identity of ions. We can do that at the level of atomistic simulations, depending on the way we perform those simulations. So what we can do is map the observations of the higher level resolution into the lower one implicitly or phenomenologically. But at the moment, that's an open field, like how to describe properly ions, not only monovalent, but also multivalent, which are very, very important for nucleosome dynamics and nucleosome interactions too, in large scale organization. Is not easy at all.
Thank you. Erin Kutz asked, in your simulations of sequence-specific nucleosome assembly, it assembles in the middle of the DNA, so presumably the consensus is in the middle. Have you tried simulations of asymmetrically DNA sequences that have longer regions on one side of the consensus?
Yes, that's something that we have done. So we have investigated, for instance, how the sequence of the DNA affects the positioning of the histone core within the sequence in our simulations, or like how different the unfolding mechanisms result if you change the sequence. And we see in our simulations, the sequence is just mapped in terms of different mechanical properties of rigidity. At the moment, all the different DNA bases have a similar strength of interactions with the different amino acids. So what we are observing now are simply effects of the mechanical properties of the DNA.
Thank you. So Chris Johnson asked, with such a challenging condensation problem, why is there so much redundant unused DNA in the genome? Do atomistic simulations reveal differences for DNA in genes as opposed to other regions, the 97% outside genes?
Yeah, so what we think that is kind of the main driving force for different types of chromatin organizations is not necessarily the length of the DNA or the sequence of the DNA, but it's more the wide range of proteins that are acting on top of the DNA. So you can have a similar DNA region, but you would have different binding affinities of proteins that have major effects in organization. So I think there's no like a contradiction between having redundant DNA regions with having a high level of organization. It's like a different set of molecules that are acting to organize the genome, in my opinion.
Thank you again. And Swati asks, lovely talk. May I know how much time does DNA take to get wrapped around histones, specifically for the first turn and second turn, according to your model?
Yeah, so in our model, when we try to just have DNA like by itself and then the histone core, we observe as in experiments that is very challenging. I don't know exactly the time that it takes. Our energy scales are not quantitative comparable to experiments. So the time that we observe in our simulation is not directly mapped to actual experimental times. But what we do observe is that forming nucleosomes just with like a DNA strand and a histone core is very challenging. So that would explain why you need additional enzymes to help in the process. We observed, however, as also the experiments have observed, that if you apply torsion to the DNA, you can favor the formation of nucleosomes, either nucleosomes or ribosomes, for instance. And we know that that also depends on the rigidity of the DNA that you are dealing with. So I don't know on the time scales that it takes, but definitely once a few contacts have been made in the right place, then nucleosomes form very, very readily. So I think it's easier to form the second turn than the first turn, but I don't know in quantitative terms what's the difference.
Thank you very much, Rosana. In the interest of time, I will let you answer the remaining questions directly on the Q&A window.
Okay.
And,
thank you.
So thank you very much, Rosana and Petra, for two very interesting talks.